Hello, my name is Olivia and I'm a postdoctoral scientist in Professor Janet Shipley's laboratory at the Institute of Cancer Research, London. I'm investigating the potential for a test known as a liquid biopsy to help improve the management and outcomes of children with rhabdomyosarcoma, and my research is generously funded by Alice's Ark. In this video, I will give you an overview of liquid biopsies and explain how we are hoping to apply them to rhabdomyosarcoma patient care.
A liquid biopsy is a sample of fluid from the body, such as blood or urine, that contains biological markers released by tumour cells. For example, as tumour cells die, they shed fragments of DNA known as circulating tumour DNA into the bloodstream. Whole cells can also break off from the tumour mass and make their way into the circulation, where they become circulating tumour cells. Scientists can use the genetic information encoded in these tiny markers to gain an understanding of how the tumour is growing or responding to treatment on a molecular level. This can include the identification of DNA mutations which could be targeted by specific drugs, or those which might indicate tumour resistance to a particular therapy. Liquid biopsies may be utilised in all stages of cancer patient care. They could be applied as a screening tool to aid in early diagnosis, a prognostic tool to help guide patient risk stratification and treatment selection, a monitoring tool to track a patient's response to treatment, or a surveillance tool to assist in early detection of relapse. Research to ascertain their suitability for each application in rhabdomyosarcoma is ongoing and will be linked to the new clinical trial for rhabdomyosarcoma known as VAR-RMS. One advantage of liquid biopsies is that they provide a specific snapshot of all the cells releasing tumour signals at the time of blood collection. This contrasts with tissue biopsies, which might only capture a small sample of a tumour mass. Liquid biopsies are also likely to be more sensitive at detecting very low levels of disease compared to techniques like medical imaging, which can detect a tumour mass down to 5 millimeters in size. This equates to around 10 million cells, whereas it is possible to detect signals from less than 100 cells per blood tube. Furthermore, liquid biopsies are incredibly safe, as a child does not need to undergo general anesthesia for blood to be collected, as is often the case for tissue biopsies and some scans. This means that liquid biopsies can enable repeated serial sampling, which can allow the patient's response to treatment to be monitored more closely than would otherwise be possible.
At the ICR, we are employing circulating tumour DNA analysis to detect treatment failure and disease relapse at an earlier stage than is currently possible, as these are major indicators of poor outcome in rhabdomyosarcoma. Using the latest genetic screening technologies, we are assessing patient blood samples collected during treatment and whilst in remission for DNA changes that we previously identified in the patient's tumour. We have just completed a pilot study to establish the feasibility of this approach and are now testing samples collected from patients enrolled on the VAR-RMS clinical trial. This is expected to lead to new tailored treatment options and improved outcomes for rhabdomyosarcoma patients. We are encouraged by the results we have achieved so far and are looking forward to continuing our partnership with Alice's Ark for the next phase of our liquid biopsy research. On behalf of all scientists at the ICR, I'd like to thank you all for your support of our research and encourage you to continue raising awareness of childhood cancers like rhabdomyosarcoma.